Assalamu alaikum. Our topic is quality control interview questions. Today we will discuss more than 110 commonly asked interview questions with answers. We will discuss basic definitions and techniques which are used in quality control, and interviewer asks questions from these techniques. We will also discuss interview questions related to different procedures and instruments which are used in quality control like HPLC, Karl, Fischer, titration techniques, infrared spectroscopy, FTIR. We will learn interview questions regarding dissolution, disintegration. All will be in this video. In this video, I will not use whiteboard because writing the answers of such a large number of questions on board is not possible. So I will use mobile phone to read all the answers with precise definitions. So let's start our first question. The first question asked is, what is quality control, or define quality control? So the precise definition of quality control is, the quality control according to WHO is defined as the collection of all the procedures which are adopted to ensure the identity and purity of pharmaceutical products is known as quality control. The question number 2 is, what is GLP? GLP stands for Good Laboratory Practices, and it is defined as a set of standard guidelines or regulations which are used to generate reproducible results which can be validated and replicated in experimental conditions. The question number 3 is, what is SISPQ? SISPQ. SISPQ is an abbreviation, and S stand for safety, I stand for integrity, S stand for strength, P stand for purity, and Q stand for quality. SISPQ. What is reference standard? Most commonly asked question. Reference standards are authentic substances of high purity and are provided by the pharmacopoeial bodies. Reference standard is also known as primary standard. So the next question is, what is working standard? A working standard is a standard that is prepared and qualified against a pharmacopoeial reference standard. A working standard is a standard that is prepared and qualified against a pharmacopoeial reference standard. Basically, working standard is used for routine testing or analysis. The next question is, why we use working standard? The answer is, the reference standard provided in a very small quantity and it is very costly, so it is not possible to use reference standard for testing all the products. That is why we use working standard, and it is prepared in-house as compared to the reference standard. Next one is the storage of working standards. The working standard after preparation are stored in a suitable bottle or vials and include following information: name of compound, physical description, LOD, assay, date of preparation, vial number, retest date, and storage conditions. What should be the ideal quantity of retention samples? The ideal quantity of retention samples should be double of those which is required to perform all the tests to indicate that API meets its defined specifications. What is incident? Commonly asked, what is incident or define incident? Any unplanned activity which results in non-compliance to the designated system or procedures at any stage of testing or storage of drug products due to system failure, breakdown, or manual error. You can learn definition by watching this video again and again. I will also provide the link of these questions in description, so you can also read all questions with answer in your free time. What is deviation? A deviation is undesired event that is different from standard or approved procedures, processes, instructions, or specifications. What is OOS or OOS? What is OOS? OOS or OOS is out of specifications and a commonly used term in pharmaceutical industries. Is defined as when the testing results are different from specifications mentioned in SOP or official compendia. What is OOT or OOT? OOT is out of trend and it is defined as the test results obtained may be within the specified limits but show a difference from historical values or results. What is calibration? Calibration is performed activity that demonstrates that the results produced by a device or an instrument are within the defined range as compared to standards. What is corrective and preventive action? So first, what is corrective action? An action that is performed to eliminate the cause of a present deviation, problem, or incident to prevent its reoccurrence is known as corrective action. What is preventive action? An action that is performed to eliminate the cause of potential deviation, problem, or incident to prevent its reoccurrence is known as preventive action. Next one is interviews question related to disintegration, and during disintegration the commonly asked question is which chapter of USP deals with disintegration. So the answer is chapter number 701 of USP, Journal chapter 701. What is disintegration test? The test which is performed to determine that how much time is required to break a tablet into small fragments which pass through mesh number 12 leaving behind mesh number 10 leaving behind no solid mass is known as disintegration test. If you want to know complete about disintegration test, the link is video is given in description, you can watch. The next is regarding the disintegration time of various tablets and capsules, and you must know the answer. So the first question will be what is the disintegration time of core tablets or uncoated tablets? The answer is not more than 15 minutes. For film coated tablets, not more than 30 minutes, and for enteric coated tablets, these should remain stable for 2 hours in acidic media and should disintegrate within 1 hour in basic media. For sugar coated tablets, not more than 60 minutes. For effervescent tablets, not more than 5 minutes. For dispersible tablets, not more than 3 minutes. For sublingual tablets, not more than 4 minutes. For filled hard gelatin capsules, not more than 30 minutes, and for soft gelatin capsules, not more than 30 minutes. So the next question is what is the temperature requirements for performing disintegration test? 37 plus minus 2. How many stages of disintegration test? There are 2 stages of disintegration, L1 and L2. How many tablets or capsules are selected for disintegration? We select 6 tablets for L1 stage, and for L2 stage we perform test with 12 tablets in 2 turns. What is the acceptance criteria for disintegration? For L1, all the tablets should disintegrate within the specified time. If 1 or 2 tablets fail to disintegrate we will repeat the test with double number of tablets, and 16 out of 18 tablets should disintegrate within specified time intervals. Why we use this skin disintegration test apparatus? To avoid the floating of dosage form. The next questions are related to the dissolution. So what is dissolution test? Dissolution is a chemical test. It is also asked dissolution is chemical test or physical test. Disintegration is physical test, dissolution is chemical test. Dissolution is chemical test that is performed for various dosage forms to check that the active pharmaceutical ingredient or ingredients are released properly and dissolved in liquid media from a dosage form or not according to the defined time limits. Why we perform dissolution test? To check that the active is released properly and is available for absorption. How dissolution test is performed? Dissolution is performed using a defined dissolution apparatus in which dosage form is dissolved in a medium at specified temperature, and samples are tested using HPLC or ultraviolet visible spectroscopy. Name different apparatus used for dissolution. Commonly asked question. 7 apparatus are used. We will learn one by one. First is basket apparatus, second is pedal apparatus, third one is reciprocating cylinder, fourth is flow through cell, fifth is pedal over disc, sixth is rotating cylinder, and 7 is reciprocating holder. What is Q value in dissolution? Most important question, what is Q value? Q value in dissolution represent the amount of API dissolved in a specified time period. Mean amount of active pharmaceutical ingredient dissolved in a specified time period is known as Q value. It is mentioned as percentage of label claim. The video may be lengthy, but these questions are helpful, so do not skip the video. Press subscribe button if you have not subscribed, and do like the video because I am recording this video for you and likes are motivation. How many stages are there in dissolution? There are 3 stages of dissolution: S1, S2 and S3. Next one is acceptance criteria for dissolution. Basically, it is a very lengthy answer. You can read the answer of this question from the link given in description. So we will move toward next question because it contains S1, S2 specification for all these 3 stages are mentioned in this answer. So do read the answer from the article given in description. What is pH? A negative log of hydrogen ion concentration is known as pH. Define pH table. pH table have value from 0 to 14 and values less than 7 represent acidic solution. Values above 7 show basic solution and middle value of 7 indicates neutral solution. What is pKa? pK value indicate the strength of an acid. The pK value is negative log of acid dissolution constant. The lower value of pKa show that the acid is stronger. Define acid. The substance which release hydrogen ion is known as acid. Define base. The base is a substance that has the ability to accept hydrogen ions. What is molarity? If you are feeling bored, do press subscribe button. Have some relaxation. You can pause the video because the video is lengthy, I know, but these answers are very important. What is molarity? Molarity is defined as number of moles of a substance per liter of solution is known as molarity. It is expressed by capital M. What is molality? Number of moles of a solute per kilogram of solvent is known as molality. It is represented by small m. What is normality? Number of gram equivalent of solute present in 1 liter of solution is known as normality. What is sonication? Sonication is a process in which undissolved particles are made soluble by degassing. We are going towards more interesting questions. We will discuss now regarding HPLC, FTIR, Karl Fischer. For do not go anywhere, just stay with the video. Titration interview questions. What is titration? Titration is most commonly used quantitative analysis laboratory method where the concentration of unknown solution is measured by reacting with the slow addition of a solution of known concentration. What is titrate? Commonly asked definitions. What is titrate? Solution of known concentration in titration is known as titrate. What is analyte? Solution of unknown concentration in titration is known as analyte. What is titration volume? The volume of titrant which neutralizes the analyte is known as titration volume. What is Erlenmeyer flask? Erlenmeyer flask is basically other name of conical flask. How titration is performed? You can read from the article, link is given in description because it is a theoretical method. Which indicators are used in the titration process? Commonly used indicators are phenolphthalein and methyl orange. What is color of phenolphthalein in acid and base? Very important question. The answer is phenolphthalein is colorless in acid. Phenolphthalein is colorless in acid and changes to pink in basic solution. What is color of methyl orange in acid and base? Methyl orange is deep red in acid and changes to yellow in basic solution. What are the types of titration? Acid-base titration, complexometric titration, redox titration, precipitation titration. Which indicators are used in complexometric titration? Starch and EDTA. It is a very most important question which is asked. Which chapter of USP deals with Karl Fischer? Karl Fischer is dealt in the chapter USP chapter number 921. What is the difference between LOD and water content? Do read the answer from the link given in description because it is a theoretical answer. This question is asked, what are the types of Karl Fischer titration? Volumetric and colorimetric. There are two types of Karl Fischer titrations. Difference between volumetric and colorimetric, read it from the link given in description. Karl Fischer works on which principle? It works on the principle of titration. Which type of titration is performed in Karl Fischer? Redox titration. Now it is very important questions regarding the components of Karl Fischer. One of the most widely and commonly asked questions. Karl Fischer is composed of following four components: iodine, sulfur dioxide, a base, methanol, and pyridine or imidazole. Why is pyridine not used in Karl Fischer nowadays? Pyridine is carcinogenic. That is why it is not used nowadays and it is replaced by the imidazole. Drift in Karl Fischer titration method. What is drift in Karl Fischer? The background environmental moisture detection is known as drift. What is used for Karl Fischer standardization? DST or disodium tartrate dihydrate is used. Why is disodium tartrate dihydrate is used? It is the primary standard, it is more stable, it is non-hygroscopic, its stoichiometric water content value is 15.66. What are the components of Karl Fischer apparatus? Detector, electrode, anode chamber, cathode chamber, rotor, electrode, drying tube, drying pipe, KF reagent, injection port, and magnetic stirrer. Which electrode is used in Karl Fischer? A platinum electrode is used in Karl Fischer titrator. Brands of Karl Fischer instrument. It is commonly asked that tell me about some brands of Karl Fischer apparatus. So Metrum KF or Mettler Toledo KF. These are two commonly used brands of Karl Fischer. Now HPLC, one of the most favorite topic of interviewers. So which chapter of USP deals with HPLC? Chapter 621 of USP deals with the HPLC. HPLC abbreviation is high performance liquid chromatography. So what is chromatography? Chromatography is a separation technique in which components of a sample are distributed between two phases. One is known as stationary phase and other is known as mobile phase. Next commonly asked question, types of chromatography. So types of chromatography are paper chromatography, thin layer chromatography, column chromatography, gas chromatography, liquid chromatography. What is chromatogram? The graph or plot obtained after completion of the separation process in chromatography is known as chromatogram. What is HPLC? As we discussed previously, high pressure liquid chromatography or high performance liquid chromatography. Components of HPLC apparatus commonly asked and most important question: mobile phase reservoir, pump, injector, column, detector, and data collection device. What is stationary phase in HPLC? Stationary phase in HPLC is the tightly packed material where absorption takes place. It does not move. What is mobile phase? As the name indicates, it is the moving phase, and mobile phase is a solvent or mixture of solvents that move during the separation process and transport solute through the column. Types of liquid chromatography: normal phase liquid chromatography, reverse phase liquid chromatography. What is normal phase liquid chromatography? The type of chromatography where stationary phase is more polar than mobile phase is known as normal phase chromatography. What is reverse phase chromatography? Liquid chromatography. Type of chromatography where mobile phase is more polar than stationary phase is known as reverse phase chromatography. This difference is commonly asked. So remember. What is column of HPLC? Column of HPLC is a structure made of stainless steel in which stationary phase is packed. Next question is commonly used stationary phase of HPLC. Silica gel is commonly used stationary phase of HPLC. Why it is used? Because it is an inert and does not react with any mobile phase. What is gradient illusion? What is gradient illusion? The technique where composition of solvent or mobile phase remains continuously changing during separation process is known as gradient illusion or solvent programming. It is also known as gradient trend. So commonly asked question is what is gradient trend? So the technique where composition of mobile phase remain continuously changing. What is isocratic run? The technique where composition of mobile phase remains same during the separation process. Mean in gradient illusion, the composition of mobile phase remain continuously changing, and in isocratic run it remains same. Various abbreviations used in HPLC: VM, TM, VR1, TR1, VR2, TR2, WH, WI, H, H/2, all these are given in the article. Kindly read from there. What is retention time? The time spent by a component in a column or with a stationary phase after sample injection is known as retention time. Which gas is used in gas chromatography? Commonly used gases are helium, nitrogen and hydrogen. Commonly asked most important questions about the detectors in HPLC. Following detectors are used in HPLC: PDA detector, UV detector, light scattering detector, conductivity detector, fluorescence detector, refractive index detector. What is used for HPLC calibration? Caffeine is used. Polarimeter interview questions. What is polarimeter? Polarimeter is an instrument that is used in quality control lab to measure the polarization of light for finding the effect of substance on rotating the plane of polarization of light. Which chapter of USP deals with polarimeter? Chapter 781. What is light source in polarimeter? A sodium lamp is used as a light source in polarimeter. Why sodium lamp is used? Because it is monochromatic and has high energy output. What is enantiomer? The mirror image of a compound is known as enantiomer. What is dextrorotatory? A compound that has a property to rotate the plane polarized light to right or clockwise is known as dextrorotatory. What is levorotatory? A compound that has a property to rotate the plane polarized light to the left or anti-clockwise is known as levorotatory. Spectroscopy interview questions. What is spectroscopy? Spectroscopy is the study of spectra produced by matter due to absorption or emission of light and other radiations. What is UV visible spectroscopy? UV visible spectroscopy is technique that is used to measure the quantity of an analyte based on the amount of light received by a sample. What is Baer law? The amount of light absorbed is proportional to the concentration of the solution. What is Lambert law? Lambert law states that absorption is proportional to the path length. What is UV visible range? The ultraviolet visible range is 200 to 800 nanometer. The UV range is 200 to 400 nanometer. Visible range is 400 to 800 nanometer. What is cuvette? Cuvette is a small sided clear container that is used for placing sample in spectrophotometer. Types of cuvette for spectrophotometer: optical glass, UV quartz, IR quartz and sphere. Which cuvette is used for UV and visible range? For UV scanning, quartz cuvette is used because it does not absorb light. For the visible range, glass cuvette is used. Components of UV visible spectrophotometer: light source, monochromator, sample holder, detector and recorder. Which light source is used for the spectrophotometer? Deuterium lamp, halogen lamp, xenon lamp, low pressure mercury lamp. Which light source is used for UV and visible range? Most important question. Deuterium lamp is used for UV range. Halogen lamp is used for visible range. Which light source is used for both UV and visible range? Xenon lamp is used for both UV and visible range. What is FTIR? FTIR is Fourier transform infrared spectroscopy and is used for the identification of substances. So we discussed more than 110 questions which are commonly asked in interviews with detailed answers. If you have any confusion, you can ask in the comment box. The video was lengthy, but I tried to deliver you the precise definitions and procedures regarding the questions which are asked. You can read all these questions from the link of article given in description. You can also visit my website pharmainform.com for more articles and questions. And if you like the video, do subscribe channel and like the video and share it to the others. Till the next video, Allah Hafiz.